Hi everyone, let's go ahead and get into Unit 17 where I want to talk about the trematodes. I'm going to first talk about digenea, which are the flukes, the lung, liver, and gastrointestinal flukes, and then I'm going to talk about aspidogastria, which are the blood and bladder flukes or the schistosomes. So, humans are the definitive host in all of these except for the sheep liver fluke, which is an accidental host. The World Health Organization estimates that approximately 30 million people have a fluke infection. Trematodes are not often seen in this country because they do require a highly specialized intermediate host that are not actually found in this country. So let's go ahead and get started. Some characteristics of flukes is that they are flattened and non-segmented. They're kind of leaf-like. They are small and only a few millimeters to several centimeters in length. They have oral and ventral suckers that are present, and these are their modes of attachment. They have a simple digestive system with no anus. They usually regurgitate their waste. Some are unisexual and some are hermaphroditic. The body of the male wraps around the female and they coexist as a pair their entire adult lives, and that occurs in the schistosomes. Some are intestinal and organ flukes and others are blood flukes. Let's take a look at the scientific names versus the common names, and you do need to know both. Phaseolepis busci is the giant intestinal fluke. Heterophes is the heterophid fluke. Fasciola hepatica is the sheep liver fluke. Clinorchus sinensis is the Chinese liver fluke. Paragonymous westermani is the oriental lung fluke. Schistosoma japonicum is the oriental blood fluke. Schistosoma mansoni is Manson's blood fluke, and Schistosoma hematobium is the bladder fluke. Okay, the lung, liver, and gastrointestinal flukes are almost always operculated and are hermaphroditic, whereas the schistosomes are not operculated and they are either males or females. The operculum is an escape hatch in the shell where the microcidium or the free-living larva escapes. That is also known as the feeding larva. The life cycle of the trematodes are pretty complex, but I do want to go ahead and get into a general life cycle of the intestinal and organ flukes. Make sure you know the intermediate hosts for all. Also know the scientific and common names. Also know the diagnostic and infective stages, and be able to recognize images of these flukes in the ova. Okay? In the generalized life cycle of the intestinal organ flukes, the adult flukes lay eggs that exit the human host via feces, urine, or sputum. That's the diagnostic stage, finding the ova in the feces, urine, or sputum. Inside the ova, there is a miracidium larva that develops and hatches. The miracidium larva penetrates ova and enters the first intermediate host, and that's going to be a snail. The larva further develops into a cercarier larva inside the snail and then emerges from the snail. The larva emerges from the snail and enters the second intermediate host, which is some sort of a plant, fish, crab, or crayfish. The human then eats the plant, fish, crab, or crayfish and ingests the encysted larva. The larva matures in the human intestine, liver, or the lung, and that is the infective stage. Let's take a look at the general life cycle of the blood flukes and the bladder flukes. These are the schistosomes. There's a nice image of the life cycle in your textbook. The ova leaves the host via feces or urine, and that's the diagnostic stage, finding the ova in feces or urine. The miracidia larva hatch from the ova in fresh water. The larva enters the snail, and the snail is the intermediate host. The larva then leaves the snail after further development and is a free-swimming larva. This larva penetrates the skin of the human host, and that's the infective stage. A schistosomule circulates and develops to maturity in the blood vessels near the intestine or urinary bladder. A schistosomule is a migratory larva in the definitive host. Some flukes, like the schistosomes, only have one intermediate host, and that happens to be the snail. The GI and organ flukes have two intermediate hosts. Usually that is the snail and some sort of aquatic vegetation, fish, or crustacean. Let's go ahead and summarize the intestinal and organ flukes. The intestinal flukes includes Phaseolepis busci and Heterophes, and you can see those pictures in that respective order on the bottom left. The liver flukes would be Fasciola hepatica, which happens to look exactly like Fasciolepis busci. The ova of Fasciola hepatica and Fasciolepis busci look exactly the same. Then another liver fluke is Clinorchus sinensis, and you can see that in the third image on the bottom. And then finally, the lung fluke is Paragonimus westermanni, and that is a ova of Paragonimus westermanni on the bottom right. Okay, now Fasciolepis busci is sometimes seen in pigs, and then in heterophyes, humans are usually an accidental host from contact with fish-eating mammals. In Fasciolepis busci, the adults live in the vessels that actually surround the small intestine. The diagnostic stage is when we see ova in the fecal exam. The first intermediate host is when the larva is hatched from the ova and enters the snail, so the snail is the first intermediate host. The second intermediate host is cercaria larvae that emerge from the snail and attach to aquatic vegetation. The cercaria larvae are free-swimming larval stages and the non-feeding stage of the larva. Some aquatic vegetation which is then eaten by humans would be bamboo shoots or watercress. The infective stage is when the larva is eaten on the aquatic vegetables like bamboo shoots and watercress. Let's talk about some specific details on heterophyes. The adults live in the vessels that surround the small intestine. This is why we call this a small intestinal fluke. The diagnostic stage is when the ova are seen in the fecal exam. The first intermediate host is the snail. It's a Myristidia larva that hatch from the ova and enter the snail. Myristidia means feeding larva. The cercaria larvae develop in and then emerge from the snail. The cercaria larva is the non-feeding larva. The second intermediate host is fish that have been infected with cercaria larvae, and metacercaria larvae develop in the fish. That's the second intermediate host. The infective stage is when the contaminated fish is then eaten by the human. Fasciolepis busci is one of the largest ova that you're going to see. It is indistinguishable from Fasciola hepatica. It is a large ova at 90 to 150 microns. It's actually very large. It is operculated, and you can see the operculum flipped open in the top image. We're unable to differentiate this ova from Fasciola hepatica because they do look indistinguishable from each other. The adult fluke has visible suckers and a uterus and testes present. Again, they are intersexed. They're usually 90 microns in diameter and about 150 microns in length. Heterophyes looks very similar to Clinorchus sinensis, and we don't want to confuse it with that pumpkin seed shape of Clinorchus. In the image on the right, we can see Clinorchus sinensis on the bottom and a Heterophyes on the top, so that's a nice way to compare the two. Heterophyes is operculated, but it has less distinct shoulders than that of Clinorchus sinensis. And there's also a terminal knob, but it is very difficult to see and almost non-existent. Heterophyes is about 15 to 17 microns in width and about 26 to 30 microns in length. In the case of Fasciola hepatica, grazing sheep and cattle seed the soil and water with ova. It is common in cattle and sheep raising areas, including the USA. Fasciola hepatica is also known as the sheep liver fluke. Ova are consumed by snails, and snails are the first intermediate host. The larvae emerge from snails, and larvae develop further to cercaria and metacercaria larvae on the watercress or water grasses. Watercress and water grasses are your second intermediate host. Water plants are eaten by the human, and larvae move to the vessels around the liver and bile ducts. This would be the infective stage. Ova are then transported to feces and leave the body, and that is the diagnostic stage by finding the ova in stool. Sheep here in this circumstance is the definitive host, and the human is the accidental host. Clinorchus sinensis is the Chinese liver fluke, and this liver fluke is found in places like China, Japan, Korea, and Vietnam. Ova and feces seeds freshwater sources, and the Myristidia larvae hatch from the ova and enter the snail. The snail, again, is the first intermediate host. The cercaria larvae leave the snail and enter a freshwater fish, and that freshwater fish is our second intermediate host. Metacercaria larvae are ingested by the human in undercooked fish. By eating that undercooked fish, you will become infected. That's the infective stage. The immature fluke migrates to the vessels near the liver and bile ducts. Ova are then passed intermittently in the feces, and finding those ova in the feces is part of the diagnostic stage. Here's an image of Fasciola hepatica, and as you can see, it looks exactly like Fasciolepsis busky. You cannot differentiate the ova. It is operculated, and it is very large, at 60 by 150 microns. And we're going to be looking for that ova in the feces. That's going to be in the diagnostic stage. Clinorchus sinensis is my favorite trematode. It is passed very intermittently, and several stool samples have to be examined in order to find Clinorchus sinensis. It is a small ova at about 15 microns in diameter by 30 microns in length. It has pronounced shoulders and a tiny operculum. There's a small knob on the posterior end, but it's difficult to see, and it does kind of resemble a pumpkin seed. Paragonimus westermanni is the oriental lung fluke. The ova are deposited in fresh water, and they hatch into Myricidia larva, which enters the snail, and that's the first intermediate host. That's the feeding larva. This happens to be a fluke that is endemic in the Far East, Africa, and South America. The larvae develop in the snail and exit the snail as a cercaria larva. This would be the non-feeding larva. The non-feeding larvae penetrate the crustaceans, and crustaceans would be things like crabfish and crabs. This would be the second intermediate host, and they develop into metacercaria larvae there. The human then eats the infested crustacean, and that's the infective stage. Larva migrates to the vessels near the lungs and bronchioles and lay eggs. The ova are coughed up in the sputum or swallowed and passed in the feces. So here we have two diagnostic stages, by finding the ova in the sputum or finding the ova in the feces. It's interesting to note that some cultures eat a meal known as drunken crab, which is live or raw crab doused in wine. And this is one way a person can develop a Paragonimus westermanni infection. Yummy. Paragonimus westermanni has a mid to large size ova. It is usually about 48 to 60 microns in diameter and about 80 to 100 microns in length. It is operculated, and the shoulders are small and may not be very apparent. It has a thick terminal shell, and that's one way you can differentiate it from Diphyllobothrium latum, which is a tapeworm. Okay, the ova of Diphyllobothrium latum and Paragonimus westermanni are very similar, but Paragonimus westermanni has a thick terminal shell. It's usually wider towards the operculated end, and you can see that in the image on the right. Diphyllobothrium latum, on the other hand, has a thinner shell and is more oval in shape. The schistosomes are the blood and bladder flukes. The blood and bladder flukes are highly adapted to intravascular existence. The females will dilate blood vessels of the definitive host of the human and lay her eggs. The eggs are very securely attached to the host due to their spikes. The sexes are separate. The ova are not operculated, and the first intermediate host is the snail. There's no second intermediate host. The infective stage occurs by penetration of the skin by cercaria larvae. Schistosoma mansoni is found in the colon or the large intestine. This is a blood fluke that resides in the blood vessels in close proximity to the intestinal tract. It actually lives in the veins of the lower colon and lives on ingested blood. The ova will be seen in the feces. It is a large ova at about 80 by 180 microns. It has a large lateral spine, also known as a rose thorn or a kickstand. You will notice that the schistosomas are not operculated. Schistosoma japonicum is found in the small intestine and resides in the veins near the small intestine as well. This was a type of schistosome that was seen commonly during World War II in American troops who acquired this parasite in the Philippines. This is a blood fluke that lives in the vessels near the intestinal tract. The ova are seen in the feces, and it is mid-sized at about 60 by 80 microns. They are more round in shape. They may have a tiny lateral spine that is difficult to see, and you can see that in the image where the arrow is pointing. The outer coat may have a sticky appearance, and they are more round than other fluke ova. They're usually about 60 to 80 microns in size. Out of all the trematodes, the Schistosoma japonicum is probably the hardest one to identify in a concentration. Schistosoma hematobium is the bladder fluke. Adult pairs live around the urinary bladder veins and adhere to the walls of the bladder. This is also associated with cancer of the bladder. They are large ova at 80 to 160 microns, and they have a large terminal spine that is easily noted, and you can see that in the image on the right. The ova become trapped in the urinary bladder due to that spine. The ova are then shed primarily in the urine, and once in a while you might find them in the feces. And then here I just wanted to wrap up by showing you the adult schistosomes. We have Schistosoma mansoni. The cercaria larva is up on the top left image. And then keep in mind the adult schistosoma fluke is about 1 to 2 centimeters in length. There are about 90 million people that are infected worldwide with schistosomas. And that's it for Unit 17, a pretty short unit on the Digenea, which are the flukes, and the Aspidogastria, which are the schistosomes. If you have any questions, please reach out.